Imagine you're walking along the beach when you spot something big floating on the water in the distance, slowly approaching and landing on the shore. Out of it come people who look different. What we don't know yet is that eventually they will invade, massacre, and take over the land that's our home. But the deadliest thing is not their advanced weapons. It's a mysterious disease that suddenly appears. Festering skin, high fever, and within days the lives of people around us are gone. This is what the indigenous people of the Americas felt when European colonizers arrived. Not weapons, not war. What killed them was something they could not even see. Viruses that managed to wipe out up to 95% of the native population. Yeah, germs are indeed some of the deadliest killers. Since ancient times, humanity has been fighting endlessly against these evil microbes. We got caught off guard a few years back. A pandemic that claimed millions of lives left us unable to leave our homes. Cities became silent and the economy collapsed. But maybe, out there are still disease-causing germs we don't know about yet. Ones that are far more terrifying and deadly. What could they be? What is the most dangerous infectious disease? Since ancient times, we humans have been in a never-ending arms race against germs. And in this war, our frontline warriors are the scientists and researchers who study infectious diseases. Think of them as germ-fighting detectives, working tirelessly to understand our microscopic enemies and find ways to defeat them. It's a high-stakes job. One mistake and the germ lives. So how do you study something that could potentially kill you and everybody else if it gets loose? The answer? You do it in a high-security lab. That's basically a fortress against microbes. Around the world, there are secret labs designed with multiple layers of protection to keep dangerous germs sealed up tight. These labs are classified by biosafety levels from level 1 to level 4. The level of the lab depends on how nasty the germ is, how it spreads, and whether there are cures or vaccines. It's like a step-by-step security system for microbial bad guys. The more dangerous and contagious the agent, the higher the BSL number and the crazier the precautions. BSL 1 is where beginners learn the ropes with microbes that don't usually make healthy people seriously sick. Think of it as a driving course in a parking lot. Gloves, coats, and good hygiene do most of the work. Move up to BSL 2 and now you're handling things that can cause disease. So you're at type of training, locked access, and specialized hoods that suck air inward to protect your face when there's a chance of splashes of tiny airborne droplets. Crucially, negative pressure rooms are not the standard at BSL 2. That's a hallmark of the next level up. There are 2 rules. 1. Match the gear to the risk. And 2. Assume that complacency is the real enemy. BSL 3 is where the air matters. Literally. These labs deal with airborne threats, the kind you could inhale in tiny amounts and still get very thick. The doors form airlocks. The whole room runs under negative pressure, so air always flows in, not out. And HEPA filters clean the air that leaves. Respiratory protection becomes routine, and you work inside ventilated safety cabinets as if the invisible world is trying to sneak past you. During COVID-19, for example, routine clinical specimen handling could be done under BSL-2 conditions with risk-based precautions. But culturing live SARS-CoV-2 virus, the step that actually grows infectious virus, belonged in BSL-3. So that split, the aerosols versus paperwork, live virus versus inactivated, captures the whole spirit of biosafety. Understand the risk and then build a moat around it. At the top sits BSL-4, the Vault. These labs are built to handle the worst known germs. Like viruses that are highly lethal and sometimes have no widely available cure. Picture researchers in positive pressure suits that puff clean air outward so nothing can seep in. Entry and exit feel ritualistic. Layer after layer of doors. A chemical shower while you're still suited. Then a second personal shower, belt, suspenders, and another belt. Facilities like this are rare and for good reason. Running one is like piloting a submarine on the ocean floor. No room for mistakes. As of 2023, researchers tracking bees labs counted about 51 bee cell 4 facilities operating worldwide, with more under construction or planned. That number sounds big until you map a planet and then you realize just how few places can safely nick humanity's most dangerous microbes face to face. So why all the drama? Because we've seen how even one step can echo outside the lab. In 1979, a release of anthrax spores from a Soviet military facility drifted downwind and killed at least dozens of people. A disaster later pieced together from medical records and wind maps. So if two words sum up the lesson, they're human error. That's why modern high-containment labs add backup after backup, alarms for airflow, strike locks, buddy system, drills, and designs that assume something will eventually go wrong and try to keep it from hurting anyone else. So, having toured the ladder of biosecurity from Level 1 to Level 4, a question might be bubbling up. What exactly is the most dangerous infectious disease? Well, to answer that, let's step back from the lab now and look at the bigger picture of humanity's fight against infectious diseases. All these high-tech labs, spacesuit, and fancy filters we'll talk about, they exist for one purpose: so that we can learn how to defeat the microbes that threaten us. It's a costly and complex war we're waging, but it has paid off in some big victories. Yet, the war is far from over. Out in the real world, infectious diseases still kill millions every year, especially in regions where healthcare and sanitation are not readily available. So consider this, even after all our modern advances, the top infectious disease killers today are old classics. In many parts of the world, malaria continues to take countless lives annually, and tuberculosis stubbornly hangs on as a big global problem. Along with HIV, these three diseases alone still kill about 2 million people each year collectively. In sub-Saharan Africa, malaria is a daily reality, and outbreaks of Ebola and other viral fevers periodically flare up, reminders that nature's arsenal of germs is diverse and deadly. Meanwhile, new threats are emerging. Some of them are new microbes like COVID-19, but others are actually our old enemies wearing new armor. I'm talking about antibiotic-resistant bacteria. Over decades of antibiotic use, many bacteria have evolved defenses against our drugs. You may have heard of superbugs like MRSA or multi-drug-resistant TB. These germs are not necessarily more contagious or more deadly by itself than regular bacteria. But when you get an infection, the usual medicines might not work. That makes a normally treatable infection potentially life-threatening. The World Health Organization warns that antimicrobial resistance is one of the top global health threats. We are already seeing infections that used to be cured by a simple pill, now requiring last resort hospital drugs, or having no effective treatment at all. It's a scary prospect. Imagine a world where routine surgery or even a scraped knee becomes risky because of untreatable infection. So, are we doomed to be overrun by superbugs and killer viruses? Well, not so fast. Remember, it's an arms race, and we humans can be pretty crafty. Scientists around the globe are fighting back with new innovations, sometimes using nature against itself in clever ways. One fascinating new, or rather revived approach is using viruses to kill bacteria. Yeah, you heard that right. If bacteria are evolving to resist antibiotics, why not use their natural predator, the bacteriophage? Bacteriophages, or simply phages, are viruses that infect bacteria exclusively. So they're like tiny guided missiles that seek out bacterial cells, inject their genetic material, and burst the bacteria from within. Phages are everywhere in nature, and each phage usually targets a specific type of bacteria. Scientists have known about phages for a century and even used them to treat infections before antibiotics were discovered. Now, with the antibiotic crisis, phage therapy is making a comeback. In recent years, there have been extreme cases where patients with superbug infections were cured by experimental phage treatments, essentially a tailored virus cocktail that killed the bacteria doctors could not kill otherwise. The cool part is, researchers can now genetically engineer phages to make them better infection fighters. Thousands of phage types exist, and using them as is can be tricky. But with new gene editing techniques, scientists can tweak a phage's DNA to, say, arm it with stronger killing genes, or alter it so it only attacks a very specific bacterial strain, leaving your good bacteria alone. In 2024, a team developed a method for rapidly editing phage genomes in large numbers, essentially creating custom-designed phages on demand. These could lead to personalized phage therapies. Imagine sending the lab a sample of your infection, and they program a virus specifically to obliterate that infection and nothing else. It's like hiring an assassin who only takes out the bad guys and self-destructs when the job is done. They're also leveraging technologies like CRISPR, a gene editing tool taken from a bacterial immune system. CRISPR can be used to detect germs with extreme precision. For example, new COVID rapid tests use CRISPR to light up if the virus RNA is preserved, and it can even directly attack viral genomes. Some researchers are exploring CRISPR-based drugs that could cut HIV out of infected cells' DNA, or chop up antibiotic resistance genes in bacteria. And let's not forget vaccines, one of our strongest weapons against infectious disease. The COVID-19 vaccines were developed in record time using mRNA technology, which itself was built on decades of scientific groundwork largely done in those labs you talk about. We now have experimental vaccines for HIV in trials. New malaria vaccines are being rolled out, and we even work on a universal flu vaccine that could cover a broad spectrum of influenza strains at once. Vaccines train our immune system so that when the real germs show up, our body soldiers, the white blood cells, recognize it immediately and destroy it before we get seriously ill. It's like showing your immune system a wanted poster of the bad guy in advance. So, which is the most dangerous infectious disease? Is it some exotic virus like Ebola? A stealthy pandemic flu? A superbug that laughs at our antibiotics? Well, in truth, it's hard to pick just one, because danger can be measured in different ways. Ebola is extremely lethal, but it usually burns out in remote areas and doesn't spread as widely. A new influenza strain might be only moderately deadly per infection, but if it infects billions, it could kill more than Ebola ever would. An engineered superbug could theoretically be the worst, but thankfully, we haven't seen a truly doomsday virus emerge yet. What's certain is that we have to stay on guard on all these fronts. Nature's germs in those we might inadvertently create. The ongoing battle between humans and microscopic organisms will likely never end. Germs constantly evolve, and occasionally jump species or develop new tricks, and we have to be ready to counter them. Given this tug-of-war, who do you think will win in the long run? Us or the germs? It's a bit of a sobering question. Bacteria and viruses have been here billions of years before us and can adapt at lightning speed. But humans have ingenuity and the tools of science on our side. It's an arms race where stopping for a moment can put us behind. Let's time travel to 1854 London. People are dying from violent diarrhea and vomiting. The dominant theory of the day blames bad air. But then, a young doctor named John Snow is unconvinced. He wields a map of death and sees a tight cluster around one water source, the bird strip bomb. Snow persuades local officials to remove the handle, and after that, cases palm up in that neighborhood. Later investigations found sewage had leaked into the well. The move did not magically end cholera, the mindset did. He had a question, a map, and the guts to test a hypothesis in the real world. If there's a lesson, it's that the deadliest infectious disease is the one we underestimate, the one we allow to sprint through gaps in vaccination, the one we let evolve under the pressure of half-finished antibiotics, the one we try to study without building a culture of safety strong enough to catch the everyday mistakes. It's also the disease we miscommunicate, where rumors outrun facts and fear defeats cooperation. Germs evolve fast, but people can too, if we decide to.